So, continuing with our discussion on enzymes, we saw that enzymes are biological catalysts which enhance the rate of reaction and make a reaction which is possible on paper practically possible. An example of this is the enzyme orotidine phosphate decarboxylase, which enhances the rate of reaction 10 raised to 17 times. Can you imagine? It's like if the reaction without the enzyme would take 78 million years or 7.8 crore years, with the enzyme, that reaction happens in a few milliseconds. That is the meaning of 10 raised to 17 times rate enhancement. Some enzymes are active on their own, just as pure proteins. But some enzymes have an additional component called as cofactor, which are molecules that either enhance the activity of the enzyme or are essential for the activity of the enzyme. That is, the enzyme cannot function without the cofactor being attached to it or the cofactor being present. These cofactors can be organic. Organic cofactors are called as coenzymes. Inorganic cofactors can be metal or non-metal ions. Coenzyme or a metal ion which is tightly bound to the enzyme, even covalently bound, is called as a prosthetic group. So it is called as a prosthetic group only when it is tightly bound to the enzyme. Protein part of the enzyme is called as apoenzyme, and so you can say that the whole enzyme is apoenzyme plus the coenzyme or cofactor. Some enzymes are modified covalently by phosphorylation, glycosylation. These alterations are involved in regulation of enzyme activity. In the sense, you will study or you will have studied that some enzymes are activated by phosphorylation, some others are inactivated by phosphorylation. So these changes are involved in regulation of enzyme activity. Coenzymes are low molecular weight organic substances. They are heat stable, very important to note, as opposed to enzyme, which is the apoprotein apoenzyme, which is thermolabile, and they are generally loosely bound to the enzyme, can be separated by the protein part of the enzyme by dialysis. This is a similar process to the dialysis that we do for patients of renal failure. There is a semi-permeable membrane and coenzyme with a dialysate are put in that. The membrane has pores with the size just enough to let the coenzyme molecules pass through, but the larger protein part of the enzyme remains in the first compartment. Coenzyme molecules pass through, and thus enzyme and coenzyme molecules get separated. And once the reaction is over, that coenzyme can act as a coenzyme for another substance. So most of the coenzymes are derivatives of vitamin B complex. So nicotinamide adenine dinucleotide, NAD. Why is it NAD plus? Why do we write it as NAD plus? Because the nitrogen in NAD is in quaternary ammonium form. So the nicotinamide adenine dinucleotide is the active form of vitamin B3 niacin, and another is nicotinamide adenine dinucleotide phosphate, NADP. And so this NAD, this acts as a hydrogen acceptor or electron acceptor or electron donor, and this NAD is the one who gives electrons to complex 1 of electron transport chain. And then those electron transport chain, the electrons will flow from complex 1 to 3 to 4, and that will result in uh, the proton gradient being formed across the mitochondrial membrane, and the protons will flow along their concentration gradient via the ATP synthase to give rise to ATP. So we all know in this reaction of glycolysis that glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase, where NADH is formed. This NADH can then enter the electron transport chain, or if there is hypoxic condition, oxygen availability is low, then pyruvate which is formed at the end of glycolysis gets converted to lactate by pyruvate dehydrogenase. And this NADH which is formed in this glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate reaction gets converted back to NAD. That is how glycolysis can continue to take place in hypoxic condition, because in hypoxic condition NADH cannot uh, give its electrons to electron transport chain because there is no oxygen available as the final acceptor of electrons. And so one of the ways to regenerate NAD so glycolysis can continue is this conversion of pyruvate to lactate. So in this reaction, NAD is acting as a coenzyme for glyceraldehyde 3-phosphate dehydrogenase. Once that is done, it also acts as a coenzyme for pyruvate dehydrogenase. Most of the coenzymes are derivatives of vitamin B complex. Organic cofactors, coenzymes, are derivatives of vitamin B complex. We'll see in further detail in the next slide. Coenzymes can be grouped into those that accept or donate hydrogen atom or electrons, which are mainly derivatives of vitamin B2, riboflavin, that is flavin adenine dinucleotide, flavin mononucleotide. Derivatives of vitamin B3 are nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate. Why do we write NAD plus NADP plus? Because nitrogen in these compounds is in quaternary ammonium state. Second group of coenzymes is those that transfer groups other than hydrogen, which are the remaining B complex vitamins and adenosine triphosphate. Can you imagine? Adenosine triphosphate is also a coenzyme which catalyzes transfer of phosphate groups. Second and third phosphate bond in adenosine triphosphate are high energy bonds. What are high energy bonds? They say that if that bond, when the bond breaks, if the energy released is more than 7.3 kilocalorie, then that is a high energy bond. That is the energy which is released when ATP is broken down to ADP. 7.3 kilocalorie energy is released. ATP is said to be high energy compound. Any molecule, any bond after breaking, if that produces more than 7.3 kilocalorie energy, it is called as a high energy compound. And any compound which doesn't have bonds which after breaking will release more than 7.3 kilocalorie energy, or those bonds will release less than 7.3 kilocalorie energy, they are called as they are not called as high energy compounds, or they are not called as high energy bonds. So ATP is like a threshold to be called or labeled as a high energy compound. And adenosine triphosphate is a coenzyme. Other coenzymes are thymine pyrophosphate of vitamin B1, pyridoxal phosphate of vitamin B6, biotin B7, coenzyme A, B5, tetrahydrofolate B9. You can see the groups that they are transferring. Acyl group, I was not taught that in my 12th chemistry. Acyl is RCO, that is a functional group which is formed after removing hydroxyl from organic acid. Acetyl is also an example of acyl group. Inorganic cofactors are metal, non-metal ions, which are essential for or enhance the activity of the enzyme. When the metal ion is tightly bound to enzyme, it is called as metalloenzyme. So here is a list of cofactors and the enzymes for which they are cofactor. Carbonic anhydrase, carboxypeptidases, alcohol dehydrogenase have zinc as the cofactor. Hexokinase, phosphofructokinase, enolase, pyruvate kinase have magnesium as cofactor. Most of the glycolytic enzymes we can see have magnesium as cofactor. Pyruvate kinase also has potassium as cofactor. Manganese is a cofactor for phosphoglucomutase glycosyl transferases. Copper is a cofactor for tyrosinase, cytochrome oxidase, lysyl oxidase, superoxide dismutase. Calcium is a cofactor for lecithinase lipase. Iron is a cofactor for cytochrome oxidase, catalase, peroxidase, xanthine oxidase. Molybdenum is also a cofactor for xanthine oxidase. All of these are metals. Selenium is a non-metal. Technically, it's not a metal, it's a metalloid, which means that it has properties of metal as well as non-metal. And selenium is a cofactor for glutathione peroxidase. Very important reaction for integrity of RBC membrane and to protect against free radical damage. Selenium is a cofactor for glutathione peroxidase. Chloride is a cofactor for amylase. So inorganic cofactors, inorganic non-metal cofactors, example is selenium and chloride. Metallic cofactors, examples are zinc, magnesium, manganese, copper, calcium, potassium, iron, molybdenum. So this covers our discussion on coenzymes and cofactors, and we will now proceed to complete the classification of enzymes which was left at the end of the previous video. Actually, the recording stopped accidentally after lies, and I not yet I don't yet know how to fuse two videos, so I decided to make a different video for classification of enzymes after lies, and also the fifth, sixth, and the seventh classes of enzymes. So last time we saw the IUBMB classification of enzymes, International Union of Biochemistry and Molecular Biology, which was given in um, 1964 by Enzyme Commission, which was formed by IUBMB in 1961. And according to that, we had six classes of enzymes: oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. Translocase was added in 2018. So oxidoreductase catalyze oxidation reduction reactions. Transferase catalyze transfer of groups other than hydrogen atom or electrons, which is done by oxidoreductase. So transferase does other than hydrogen atom or electrons. Then hydrolase breaks down compounds using water. More importantly, hydrolase breaks single bonds using water. So we saw the reaction of hydrolase was AB plus H2O gives AOH plus BH or AH plus BOH. So there is a single bond between AB and water becomes H and OH. H goes to one, OH goes to another. So this one compound is being broken down into two. Please keep this in mind because there is an important concept that I want to clear. Lyase does the same thing. It breaks down bond between compounds without the use of water. That is what we said. And then there is but also there is uh, it may either break down single bonds without the use of water, but there is a subclass of lyase which breaks down double bond by addition of water, or there is removal of water from the compound causing formation of a double bond between the two groups. and that is a subclass of lyase and not hydrolase, and that subclass is hydratase. Hydratase is a subclass of lyase. The difference is that hydrolase breaks down single bond using water, whereas hydratase also uses water to break double bond, and that water gets attached to it doesn't form two molecules out of one, but double bond gets broken down by addition of water, or removal of water leads to formation of double bond in the molecule. So both hydratase and hydrolase are using water to break bonds. Of removal of water will form a bond, that is also hydrolase. So, but mechanism is different. Hydrolase is acting on single bond, hydratase is acting on double bond. And then we'll see the fifth, sixth, and seventh classes of enzymes. Fifth class of enzymes is isomerase. Isomers are compounds having same number of atoms of constituent element but different spatial arrangement or bonding pattern between the atom, which causes either the compound to be different altogether, like glucose, galactose, but or it could just cause optical isomers like D-glucose, L-glucose, levorotatory glucose, dextrorotatory glucose, optical isomers, or epimers, or with totally different compounds with different chemical properties, but same number of atoms of constituent elements. All glucose, galactose, mannose, they have this formula C6H12O6, fructose, but they are different compounds. And so isomerases are enzymes that catalyze isomerization reaction. Examples are triose phosphate isomerase, which catalyzes the reaction conversion of glyceraldehyde 3-phosphate to dihydroxyacetone phosphate. Glycolysis, dihydroxyacetone phosphate. This triose phosphate isomerase catalyzes this reaction. Epimerases, racemases, these are different subclasses of isomerases. So the sixth group of enzymes is ligase, which catalyzes ATP-dependent condensation of two molecules. So can we say that ATP is a coenzyme for these enzymes? Yes, we can say that. Example is acetyl-CoA carboxylase, formation of malonyl-CoA as a first step of fatty acid synthesis. And then synthetase. If you see the if you see the name of the enzyme ending in synthetase, it means it is a ligase. That means it uses ATP to condense the two molecules or to catalyze its reaction. Carbonyl phosphate synthetase, arginosuccinate synthetase, phosphoribosyl pyrophosphate synthetase, PRPP synthetase, de novo synthesis of purines, glutamine synthetase. But synthases are not ligases. They belong to different groups like delta amino levulinic acid synthase, which catalyzes the first reaction of heme synthesis, glycine plus succinyl CoA, forming delta amino levulinic acid with the release of carbon dioxide and coenzyme A. Pyridoxal phosphate is a coenzyme for this reaction. ALA synthase is a transferase, acyltransferase. Glycogen synthase is also a transferase, glycosyl transferase. Synthases do not require ATP directly, maybe requiring indirectly, but not directly. That's why they are synthase. They do not belong to ligases because ligase is ATP dependent condensation of two molecules. And you can remember synthetase has T in it. T ATP has T in it. And synthase doesn't have T, so they don't require ATP. uh, so that finishes ligase. And then we move on to the seventh class, newly added translocase. It is a very interesting class, so we'll see about it. So translocase was added in 2018. What does it mean? Translocation is an umbrella term. It means basically just moving from one location to another. बोरिया बिस्तर उठा के एक जगह से दूसरी जगह जाना. Movement of ribosome along mRNA as it slides forward during translation of mRNA is also called as translocation. Movement of one part of substances from one part of the cell to another, or movement of nutrients across the phloem of plants, that is called translocation. In genetics, translocation is when a piece of chromosome breaks off and attaches to another chromosome, that is called translocation mutation. But translocation in terms of enzymology has a different meaning. We all know that there are transporters across cell membrane. There is active transport, passive transport. Some transporters use ATP to move ions, molecules against their concentration gradient. And some are passive transporters which facilitate diffusion along the concentration gradient. So transporter is a general term used for transporting different ions or molecules across cell membrane. But when this transport happens with the help of ATP hydrolysis, and it is also this process of transporting that substance across the membrane is also linked to some kind of a group transfer reaction, that is what is called as, then that transporter can be called as a translocase, is my understanding of translocase. That is why every active transport cannot be called as translocase, because say sodium potassium ATPase, it cannot be called as translocase because it is moving 3 sodium out and pushing 2 potassium in against their concentration gradient. And but that sodium and potassium, they are not taking part in any group transfer reaction, right? That is why it is not a translocase. However, these two examples, if you see, carnitine shuttle system, which facilitates the movement of long chain fatty acids from the cytoplasm to the mitochondrial matrix, so that the long chain fatty acid can be oxidized, beta oxidation. That is a translocase because that is moving the long chain fatty acid. It is facilitating also, it is linked to a group transfer reaction later on, where coenzyme A takes this acyl group from the carnitine and becomes acyl CoA. So effectively the acyl of the long chain fatty acid has got transported from cytoplasm to the inner mitochondrial matrix. And though this does not require ATP directly, but coenzyme A being a high energy molecule, it is utilizing ATP indirectly. So my understanding is help of ATP. ATP hydrolysis may not directly be causing this movement across translocation across the membrane, may be indirectly involved, but there is help of that involvement of ATP hydrolysis. So it is sort of an active transport, and it is also linked somewhere to group transfer. So there is also another ATP ADP translocase or adenine nucleotide transporter or translocator, which is also present in the inner mitochondrial membrane. It translocates ATP from the mitochondrial matrix to the cytoplasm and ADP from cytoplasm to the mitochondrial matrix. Why is it called as a translocation? Because this ADP will then will become acceptor of inorganic phosphorus in reactions which form ATP in the cytoplasm. And sorry, the ADP will accept phosphorus in the mitochondrial matrix. It will get phosphorylated to ATP, and ATP outside will be broken down for various reactions which are taking place in the cell for which they need ATP. So this movement across the cell membrane, which somewhere needs ATP and also is related somewhere to some kind of a group transfer, is translocase. So every active transporter cannot be called translocase. Like sodium potassium ATPase cannot be called translocase. And also all translocases are transporters ultimately, but all transporters are not translocases, because firstly there is some kind of a active transport type thing, and group transfer, it is involved somewhere, and that is my understanding of translocase. But if you want to write translocase, like what do we write in exam for translocase 7th class while describing classification of enzymes? You just write it was added in 2018 and uh, it was added in 2018 and it facilitates translocation of molecules or ions from one part of the cell, from one side of the cell membrane to another side. Translocase facilitates movement of ions or molecules from one part of the cell membrane to another part. That is it. That is all you write in the exam. Translocase facilitates movement of ions or molecules from one part of the cell membrane to another part. Rather, from one side of the cell membrane to another side of the cell membrane. And then you write these two examples. Carnitine shuttle system involved in translocation of long chain fatty acids into mitochondrial matrix, and ATP-ADP translocase, adenine nucleotide transporter, which sends out ATP from mitochondrial matrix to the cytoplasm and ADP from the cytoplasm to mitochondrial matrix. That is all that you have to write in the exam. But I was trying to understand what is this translocase. You can also write in exam later on why this translocase came into picture. And that's why I'm going to explain it a little bit more. I'm going to just indulge you a little bit more because uh, so many of our senior professors, uh, they must have spent a long duration of their career without this translocase being a part of the classification. 2018 is just 9 years ago. So that is, and what forced the IUBMB to bring about this 7th class? What was going wrong that needed to be corrected, or what was happening? I wanted to explore that a bit, and I would like to share with you what I found. Before we go to that discussion, I just wanted, for official purposes, tell you what you can write about translocase in the exam. This is what you can write. New class introduced in 2018 translocates ions or molecules from one side of the cell membrane to another. Carnitine shuttle system, which translocates long-chain fatty acids from cytoplasm into the mitochondrial matrix for their beta oxidation. Adenine nucleotide transporter or antiporter, which translocates ATP out of the mitochondria and ADP into the mitochondria. So as you can see here, on the IUBMB website, I found this when they introduced the new class in August 2018. 6 enzyme classes have been recognized, and these were based on the type of reaction catalyzed. However, it has become apparent that none of these could describe the important group of enzymes that catalyze the movement of ions or molecules across membranes or their separation within membranes. Several of these involve the hydrolysis of ATP and had been previously classified as ATPases. That means they were previously classified as transferases, although the hydrolytic reaction is not their primary function. These enzymes, so you can write in exam that the seventh class was introduced because previously these were wrongly classified. Some of them were put in ATPases, transferases, even though the hydrolytic reaction is not their primary function. And they have now been classified under translocase. Reaction catalysts are designated as transfers from site 1 to site 2 because designations in and out, again, which were used previously, they lack clarity, can be ambiguous. Comments associated with each entry then describe specific translocations catalyzed. So then the subclasses of translocase. Translocation of hydrons. Hydron being the general name for protons, H plus in its natural abundance. New information for me as well. Then translocation of inorganic cations and their chelates. Inorganic anions, amino acids and peptides, carbohydrates and derivatives, translocation of other compounds. Subclasses concern the reaction that provided the driving force for the translocation, where these are relevant, like translocations linked to oxidoreductase reactions, translocations linked to hydrolysis of nucleoside triphosphate. Um, so maybe beta oxidation, maybe this translocation of oxidoreductase, that adenine nucleotide transporter, maybe this translocation linked to hydrolysis of nucleoside triphosphate. Then or maybe this hydrolysis of diphosphate, then translocations linked to decarboxylation reaction, exchange transporters that are not dependent on enzyme catalyzed reaction, such as exchange of ions across membrane like sodium potassium ATPase. They are not included, and pores that change conformation between open and closed states in response to phosphorylation or some other catalyzed reaction are classified under isomerases. 5.6 Macromolecular conformational isomerases. So they have still those gates, no, which open and close pores. They are classified as macromolecular conformational isomerases. And uh, so that is what I found on IUBMB website. And now I am on IUBMB website. And this is what I see here when I say translocase IUBMB. List of accepted names. So these are the subclasses of translocase as we read up. And these are the, oh, so many enzymes. You can read at your leisure. So basically translocases are transporters. You can see here so many are ending with like P type sodium transporter, ABC type transporter, sodium potassium exchanging ATPase. Oh my god. Even that sodium potassium ATPase is a translocase. Oh my god. So it's really confusing. So it totally debunks my previous when I said that sodium potassium ATPase is not a translocase, but it's written here sodium potassium exchanging ATPase. So what is a part of translocase? What is not a part of translocase? Who knows? But I just wanted to. The official lecture ended long ago when we finished ligase, and I told you about what I can write, what you can write in the exam. But this is just me playing around out of curiosity. Thank you for interest, and we'll see. We'll meet again in the next talk. Thank you.